Welcome to Yilan Marine Research Station. Just take a moment with us. Picture yourself standing here, east coast of Taiwan, incredibly rich nature all around.
Mountains on one side, the Pacific on the other.
Exactly. And it really makes you think about all the, intricate life happening everywhere, much of it hidden from view.
And that idea of hidden, intricate life. It's kind of the perfect starting point for our deep dive today, actually.
Oh, yeah.
We're looking at something really tiny happening microscopically, but it's fundamental to understanding how complex organisms form. Like, many of the ones right here in this environment.
Right. And the source we're digging into today, it isn't your standard textbook, is it? Though it definitely connects to textbook concepts.
No, not a textbook. It's essentially based on a script for a video guide, a practical one.
So like a hands-on tutorial.
Exactly. Teaching a core skill, how to handle chick embryos for histological analysis. Think of it like a workshop, but, in audio form.
Okay, mastering a specific process to actually see what diagrams only hint.
That's the idea, to unlock that visual understanding.
So what's the big picture here? Why go through all this really meticulous work with a tiny chick embryo?
Well, the main goal, as the guide lays it out, is to perform histological observation, looking closely at tissues on structures you can only see during development.
Things that disappear later on.
Right, like somites, the notochord, neural crest cells, things you read about in developmental biology. The idea is to take those terms off the page.
And make them real.
Exactly. Turn them into clear, vivid, three-dimensional mental images. You want to see what they really look like, their cells, how they sit relative to everything else inside the embryo in 3D space.
Okay, moving beyond the flat diagrams, and this specific protocol, it focuses on a couple of key areas for observation.
It does, yeah. The main focus is observing somites. These are the precursors, the building blocks for the axial skeleton and muscles.
Right, the backbone and associated muscles.
And also creating tissue sections of limb buds, those little bumps that pop out on the side.
The very beginnings of arms and legs.
That's them, seeing how they start to form.
So this isn't just about looking at tiny things for fun. It sounds like there's a clear scientific objective here.
Oh, absolutely.
Yeah.
The guide is very clear. This is by no means a hobby or for aesthetic purposes. It's tied directly to understanding fundamental biology, research, education, that kind of thing.
You mentioned bridging that gap with textbooks. Why is doing this hands-on work so important for that?
Well, think about it. A diagram is static, right? It's stylized. But preparing and actually looking at these thin sections, you see the real cells.
Ah, the actual texture and density.
Yeah, the density of the tissues, the exact boundaries, how structures like the neural tube and notochord are positioned next to each other, how the somites wrap around. It makes terms like mesoderm or ectoderm mean something visually. You see what the tissue looks like at that critical moment.
Because these structures, these early patterns, they're fleeting, aren't they? They're only there for a short window during development.
Precisely. Once things differentiate or cells migrate, that initial form is gone. So this technique lets you capture that specific moment in time.
Like a snapshot of the foundations being laid.
Exactly. The initial blueprint being physically built, cell by cell, tissue by tissue. It's a direct window into how life gets constructed.
Okay, let's get into the practicalities. Where does this whole journey begin?
It starts, simply enough, with fertilized chicken eggs. The guide mentions just getting them from a local poultry farm.
Okay, keeping it accessible, then what? Preparing them for incubation?
Yeah, some basic but really crucial first steps. You got to disinfect the shells, usually with ethanol.
Standard lab practice.
Right. And then mark each egg. Give it an ID, note the date you set it, just good organization.
Makes sense. And then into the incubator, what are the conditions?
They're kept pretty warm, around 37.5 degrees Celsius. But the timing when you actually take the embryos out, that's super critical.
It's not just a set number of days.
No, not really. You use a standardized system. It's called the Hamburger and Hamilton staging table.
Hamburger and Hamilton.
Sounds memorable.
It is, and it's a universally recognized guide. It describes specific developmental landmarks in the chick embryo.
So physical features you look for.
Exactly. Things like the number of somites, how developed the optic vesicles are, the early eye structures, or even the pattern of the heartbeat. These tell you the precise developmental stage.
Ah, so it standardizes everything, even if incubation times vary slightly.
Right. It allows researchers everywhere to compare results consistently. But, working with biological material like eggs, there's always a bit of variability.
The guide mentions a challenge there.
Yeah. Fertilization doesn't always happen at the exact same moment. Development might start slightly differently, even in eggs from the same batch.
So you can't just rely on one egg.
Definitely not. The practical solution, you prepare multiple specimens, incubate several eggs. That way you boost your chances of getting at least a few embryos at exactly the stage you need. Redundancy is your friend here. Okay, incubation's done, you've got your eggs. Now the really delicate part, getting that tiny embryo out. How do you even start?
First step is carefully opening the egg. Then, the big obstacle is the albumin, the egg white. It surrounds the embryo and makes it hard to see.
Right, it's thick and sticky.
Exactly. So you need to remove it carefully. The guide mentions using a custom-modified syringe needle, bent just so.
A bit of lab hacking.
Sort of, yeah. To carefully suck out the albumin without disturbing the yolk or the embryo sitting on top.
Clever. Okay, albumin gone. But the embryo's still pale against the yellow yolk, right? How do you see it clearly?
Ah, this is a neat little trick. It's about adding contrast. You inject a diluted ink solution,
Ink, like writing ink.
specifically Sumi ink, mixed with Ringer solution, which is a saline solution. You inject it underneath the embryo, between the membrane it sits on and the yolk itself.
Oh, I see. So you create a dark background behind it.
Exactly. That dark ink layer makes the translucent white embryo pop. Suddenly it's much easier to see, to handle, to figure out its orientation.
Simple, but effective. And then actually lifting the embryo out. Does that depend on its size or stage?
It really does, yeah. If it's a bit larger, more robust, you might be able to gently scoop it out with a small spoon, then transfer it to a dish of PBS that's phosphate buffered saline, just a holding solution.
Okay, but the smaller ones.
Smaller, more fragile embryos need more care. The guide describes using filter paper. You punch a hole in a piece, carefully place the paper over the embryo.
So the embryo sticks to the paper slightly.
All right, it adheres just enough. Then you trim the paper around it and use that paper like a little raft.
Wow.
Yeah, use the paper raft to lift and transfer the embryo stably into the PBS dish using a spoon. It helps prevent it from folding or getting damaged.
That sounds incredibly precise.
Yeah.
Needs a steady hand. Okay, embryo is safely in the PBS. How do you confirm its stage then?
Now you go back to that hamburger and Hamilton table. You put the dish under a dissecting microscope and look closely.
Counting somites again.
Counting somites, looking at the eye development, those optic vesicles, checking the heartbeat pattern, all those landmarks together tell you its exact H&H stage.
Got it. So you have your embryo, confirmed it's the right stage for what you want to see. What's the next critical step to sort of lock everything in place?
You need to fix it. Fixation. It's a process that rapidly kills the cells, but preserves their structure, their components, exactly as they were at that moment.
Freezing it in time, essentially.
Pretty much. And this protocol uses a common fixative called Boone's solution.
Boone's solution. Okay. And the guide flags something important about this one.
Yes, a really crucial safety note. Boone's contain some hazardous chemicals, picric acid, formaldehyde, acetic acid.
Right. Stuff you don't want to breathe in.
Definitely not. It must be handled inside a chemical fume hood. Proper ventilation is essential. Safety protocols are non-negotiable here.
Understood. Safety first. And like getting the embryo out, how you apply the fixative also depends on the size.
Correct. Again, it's about preventing distortion. Bigger embryos, you can just immerse them directly in a vial of Boone's.
Okay.
But for the smaller, more delicate ones, especially those maybe still on their filter paper raft, you often fix them right there in the PVS dish.
How?
By gently spraying the fixative over them. This helps maintain their shape as the fixative starts to work before they get too rigid.
Locking in the structure before it can deform makes sense. After fixation, what's next?
Okay, so after fixation, the tissue is preserved, but it's full of water. And the goal is to eventually embed it in paraffin wax, which hates water. It's hydrophobic.
So you need to get the water out.
Exactly. Through a process called dehydration, you pass the embryo through a series of ethanol solutions with increasing concentrations.
Like 70%, then 80%, 90%, up to 100%?
Usually something like that, yeah.
Yeah.
Starting maybe at 70% and going incrementally up to 100% pure ethanol. This gradually pulls the water out and replaces it with alcohol.
Swapping liquids inside the cells, slowly.
That's a good way to think about it. Once it's fully dehydrated and 100% ethanol, you need something that mixes with both ethanol and the melted paraffin wax you'll use next.
A transition step.
Right. The guide mentions using a xylene substitute. Lemosol A is the example given. So ethanol out, Lemosol A in, then Lemosol A out, and you transfer the embryo into melted paraffin wax.
And the warm wax seeps into all the spaces.
It infiltrates everywhere throughout the tissue. replacing the Lemmasol A.
Okay, so the tissue is now full of liquid wax. That leads right into embedding, I imagine.
It does. The goal now is to take that paraffin-infused embryo and make a solid block of wax around it. That block is what you'll eventually slice.
How do you make the block?
You take small metal molds, fill them with fresh melted paraffin, and then place the infiltrated embryo inside. And here's a really critical part: orientation.
Ah, how you position the embryo in the wax.
Exactly. You have to think ahead. What structures do you want to see later? Which way do you need to slice through the embryo to see them properly? You orient it carefully in liquid wax.
So if you want cross sections versus longitudinal sections, you position it differently here.
Precisely. Get the orientation right. Let the paraffin cool just enough to hold it in place. And then you typically mount this mold onto a plastic cassette.
What's the cassette for?
It's just a holder, basically. It provides a standard frame that clips securely into the microtome, the slicing machine. The guide mentions a couple of ways to mount it, depending on size and goals.
All right, so we have our perfectly oriented embryo embedded solid in a paraffin block ready on its cassette. Now for the slicing, making those incredibly thin sections.
Right. This is where the microtome comes in. It's a precision instrument designed to cut extremely thin, uniform slices from that paraffin block.
How thin are we talking?
Typically around 5 to 10 micrometers thick. A micrometer is 1/1000th of a millimeter. So yeah, very, very thin.
Wow. A microtome sounds serious.
It is. And the guide brings up another absolutely critical safety warning here.
Okay, let's hear it. Essential stuff.
The blade on a microtome is unbelievably sharp, like beyond razor sharp, designed to slice cleanly through wax and embedded tissue.
Okay.
The guide strongly emphasizes, never use one without proper training from someone experienced. And always, always double-check the safety guards and mechanisms before you even touch it. It's powerful and potentially dangerous if you're not careful and well-trained.
Got it. Respect the blade. Expertise and caution are key. What kind of environment is best for actually doing the sectioning?
The guide recommends a quiet environment. When you're trying to cut slices that are mere micrometers thick, you need focus. Even small vibrations or distractions can mess up the cut, ruin your sections. Stability is important.
And the goal isn't just single slices, but ribbons.
Ideally, yes. As the blade cuts each section, the heat and maybe a bit of static electricity make the edge of one slice stick to the next one cut.
Ah, forming a continuous ribbon of sections.
Exactly. Getting a nice, long, smooth ribbon is a sign of good technique. And it's useful because having the sections in order helps you trace structures from one slice to the next. Understand that continuity.
Okay, these delicate ribbons of wax and tissue, how do they end up on a glass slide?
Carefully. You typically float the ribbon onto the surface of a warm water bath. The warmth helps relax the paraffin and flatten the ribbon out perfectly.
Removes any wrinkles from cutting.
Right. Then you carefully bring a clean glass microscope slide up underneath the floating ribbon and lift it straight out of the water. You want the ribbon aligned nicely on the slide. It takes practice.
I bet. And then the slides need to dry properly.
Yes. Usually you place them on a slide warmer or hot plate, set to maybe around 42 degrees Celsius, often overnight. This helps the paraffin melt just a tiny bit, making the tissue section stick firmly to the glass.
So they don't wash off during staining later?
Exactly. You don't want to lose your precious sections halfway through the process.
Okay, we've journeyed from egg to these slides with incredibly thin sections stuck on them. But there's still mostly transparent wax and tissue, right? You can't see much detail yet.
Not really, no. To actually visualize the cells, the tissues, the different structures, you need to stain them, add color. But first, you have to get rid of all that paraffin wax again.
Right. How do you do that without damaging the tissue slice?
You basically run the dehydration process in reverse. First, dissolve the paraffin using something like Lemmasol A again, or the traditional chemical, xylene. Immerse the slides until the wax is gone.
Okay.
Then you rehydrate the tissue sections by taking the slides back down through that graded ethanol series, 100%, 95%, 80%, 70%, down to water. The guide notes, if you don't get all the paraffin off, you'll see hazy white patches on your final slide, obscuring detail. So complete removal is key.
Right. Paraffin gone, tissue rehydrated, ready for color.
Now for the staining. Interestingly, the protocol described actually starts with a stain called Alcian blue.
Alcian blue. What does that show?
It specifically stains acidic mucopolysaccharides. These are important components in things like cartilage matrix, some connective tissues.
So it highlights those specific components in blue.
Exactly, giving them a distinct signature. And then comes the absolute classic, the workhorse stain in nearly all histology labs, hematoxylin and eosin.
Often just called HE staining or H stain.
Yep, He stain, that's what everyone calls it. You don't usually say the full names.
Okay, he stain it is. What does that combination reveal?
This is what gives you that classic, recognizable look. Hematoxylin is a basic dye, so it stains acidic things in the cell, primarily the DNA and RNA in the nucleus, a dark blue or purple color.
Making the nuclei really stand out.
Right. And eosin is an acidic dye, so it stains the basic components, mainly proteins in the cytoplasm and the extracellular matrix like collagen, various shades of pink.
Ah, so you get the blue-purple nuclei against a pink background of cytoplasm and tissue. That must make things incredibly clear.
It really does. It provides fantastic general morphological detail. You can easily see the overall tissue structure, cell shapes, how things are arranged. It's the standard for a reason before you move to more specialized stains, if needed.
So we've done it. From fertilized egg all the way to stained sections on a slide, ready for viewing. Now the payoff. Looking under the microscope.
You typically use a standard upright microscope for this.
Mm-hmm.
And interpreting what you see, that's the next crucial skill.
Right. How do you take these flat 2D slices and rebuild that 3D picture of the embryo in your mind?
Well, that's the art and science of histology. You look at adjacent sections on the slide, section after section in that ribbon. You follow structures as they appear, change shape, connect to others, and maybe disappear further along.
Mentally stacking the slices.
Exactly. You're mentally reconstructing the three-dimensional form. And the guide makes a really good point here. Remembering what the whole embryo looked like under the dissecting scope before sectioning, that gives you vital context.
Ah, having that initial mental map helps you place the 2D details.
It really does. You're not starting completely blind. You have a framework for understanding the slices.
Okay, let's talk about those specific targets again. Somites and limb buds. What do they actually look like in these HE-stained sections?
Well, if you're looking at sections through the main body, the trunk, the somites are pretty obvious. As the guide says, they look like these block-like clusters of cells, quite densely packed.
The building blocks you mentioned earlier.
Yeah, and seeing them there, so clearly organized in segments next to the developing spinal cord and the notochord, it's like seeing the literal foundations for the vertebrae and muscles being laid down.
The very start of the body plan. And the limb buds.
When you look at sections going through a limb bud, you start to see the beginnings of different tissues forming. The guide mentions observing early cartilage formation, maybe areas where cells are packing together, condensing, rounding up. That's the start of the skeleton in the limb. And you also see the precursor cells for muscle starting to organize. It really shows you how and when these distinct tissues first emerge and arrange themselves. It's a snapshot of cells making choices, deciding their fate.
That's incredible, seeing that fundamental process happen. The guide also mentions using digital images.
Yeah, capturing digital photos of the sections is pretty standard now. It lets you study them later, zoom in, and importantly, annotate them.
Labeling things directly on the image.
Right. Labeling the neural tube, the somites, different parts of the limb bud. Then you can compare your annotated pictures back to the actual glass slides. It really helps solidify your understanding of the histology, reinforce what you're seeing.
A powerful learning tool. Okay, so that really walks us through the whole intricate process, step by step, from a humble egg to these detailed, stained, microscopic views. It's quite something.
It absolutely is. And like the guide suggests, actually going through this whole hands-on process gives you a truly tangible sense of how development works. The precision needed, the visual reward of seeing clear tissue structures.
A tangible sense? Yeah, because you physically interacted with it at every stage, handling, fixing, slicing thinner than paper, staining. It makes those abstract concepts feel really solid.
It takes developmental biology out of the purely theoretical realm and makes it physical, visual. You connect with it differently.
So as we wrap up this deep dive, thinking back to where we started here at the Yilan Marine Research Station, surrounded by all this vibrant life.
Yeah, the fish, the birds, the forests.
It does feel connected, doesn't it? This very focused microscopic lab work ultimately helps us understand the bigger picture of life around us.
It really does. Because these techniques, this kind of detailed observation of how structures form in an embryo, they reveal the processes that built all these organisms we see.
It helps us appreciate life not just on the surface, but the incredibly precise, complex construction happening inside during development.
Yeah.
Seeing those somites form, seeing the limb buds emerge, it gives you a real appreciation for the sheer orchestration involved.
You realize that the adult forms we see are just the endpoint of this amazing hidden construction process.
And maybe that's the real takeaway here. Engaging with techniques like this, the ones in this protocol, forces you to observe closely, to think about organization and change on a microscopic level. It helps cultivate a genuinely three-dimensional understanding of living things.
Moving beyond just the surface appearance.
Exactly. It's a reminder that inside every creature, there's this incredible history of development, of precise construction, this deep dive into how we study the chick embryo. It's really an invitation to appreciate that profound complexity connecting the detailed work on the bench right back to the amazing natural world surrounding us here in Yilan.
Yeah, fostering that deeper appreciation for the complexity hidden within every living thing, layer by intricate layer.